Okay, hello everybody. For those of you who don't know me yet, my name is Jan Engelmann, and I'll be your lecturer for the NEUROECONOMICS course, which I'll be teaching together with Jan Hausfeld. You will get my video lectures hopefully every Thursday evening, so that gives you the weekend to look at it and the weekend to also start asking some questions about this for the Q&A, which we'll hold via Zoom on Mondays. And I'd like to do this at 11 o'clock. Well, that's actually the official time. And then we'll have tutorials Wednesdays at 11. And if you have any questions, you can email me at this email address here. So the purpose of this brief video lecture is to give you an idea of the outline of the course and how you'll be evaluated in the end. So let's get started. So in terms of the purpose, one of the goals that I want to accomplish with you guys here is I want you to be able to understand and actually reproduce a typical fMRI method section. Let me just read this to you. So for fMRI data analysis, these authors are saying pre-processing and statistical analyses of functional data were performed using SPM8. We'll be using SPM12. All functional volumes were realigned to the first volume using B-spline interpolation and were unwarped using field maps estimated by SPM to remove residual movement-related variance due to susceptibility by movement interactions. To improve co-registration, bias-corrected and co-registered anatomical and mean echoplanar images were created using the new segment toolbox in SPM. The forward deformation fields created in the context of the nonlinear normalization of individual gray matter tissue probability maps were then used to normalize the functional time series to the Montreal Neurological Institute, or MNI, T1 template. Finally, functional data underwent spatial smoothing using an isotropic 6mm full-width half maximum Gaussian kernel. Statistical analyses were then performed using the general linear model, blah, blah, blah. Okay, you can read this in your own time. But the point is that if you're not so familiar with neuroimaging methods, this course will allow you to read this and understand this and actually reproduce what I just read to you at the end of this course. So the course goal is basically to give you exposure to the most common method that's used in neuroeconomics and other neurosciences or other fields of cognitive neuroscience, affective neuroscience, as well as computational neuroscience, to give you an idea of, well, at the very least, to be able to critically interpret many of the articles that we read, many of the articles that inform our thinking that are cited and reproduced in the public press, sometimes really wrongly. And so you'll be, at the very least, a much more critical reader because you will understand the limitations of these methods that neuroscientists use. The figures that they produce always look really convincing with those blobs on the brain, and you'll be producing those figures at the end of this course. But our goal here is to give you exposure to how these methods are used, to the powers of these methods, what they can reveal about it, and maybe even identifying applications in the real world that can change society. For instance, people have been working on using fMRI as a lie detector, which could be used in, let's say, court cases for or against you, obviously. But also the use of these methodologies and knowing these limitations in contexts such as neuromarketing. So you can critically evaluate these types of methods, and that's an important thing that I want you to be able to do at the end of this course. Well, additionally, obviously, there are some much clearer course objectives that are much closer to the material, and let me just summarize those for you. So at the end of the course, you'll be able to understand the principles underlying pre-processing of fMRI data. And this will be covered in the lectures and the tutorials. Understand the principles of fMRI experimental design. Again, this will be covered both in lectures and tutorials. And implement standard pre-processing steps using the software package SPM12, which runs in MATLAB. So you need to have MATLAB installed, and I would ask you, after you see this, to go to the SPM12 webpage and also install SPM12. It's a simple toolbox that you add to MATLAB, and it's basically described in the SPM12 manual how to install this. But if you have questions, we can always help you. Then understand the principles underlying statistical analysis of fMRI data. So we will, after we've done all the pre-processing steps, we will finally go to actually analyzing the fMRI data that we give you. And you will be able to produce those blobs in the brain that you see in many articles. And then implement statistical analysis of fMRI data for individual participants, which is a first level model. And then you'll also do a second level model. So standard fMRI statistical analysis process in 2 stages. You have a first level and you have a second level model. And we'll discuss this in detail in the class. And as I mentioned already, I would really like you to be able to critically evaluate current fMRI research. So what you know about the methods, what you know about, what you will learn about the limitations of the methods that we use in neuroeconomics, should put you in a position to critically evaluate, papers that have been published, but also when someone proposes, let's say, you take on a consulting job afterwards and someone proposes, well, let's use neuromarketing. Then you can argue that maybe this might be a method that's too expensive for what it can yield. Or you can argue for it, depending on what your goals are and what your insights are into these methods that we're going to introduce here. Okay, so the structure of this course. I will upload video lectures every Thursday, so you can have a look at in your own time. From the safety of your own home, obviously. Then I'm asking you to submit at least one question about the lecture, something that can sort of garner a discussion, ideally an open question, because we will discuss the lecture then on Monday. These questions can be things that you didn't understand and would like some more information or a better explanation of if I didn't deliver a good explanation. But it could also be things that you're interested in and just want more information on or a broader perspective on. And then we can discuss these kinds of things. I'm asking you to submit your questions every Sunday on Canvas, ideally Sunday evening at 10, so that you get some sleep before the lecture and that I can have some time to look at it. And then the Q&A sessions will take place during the actual lecture hours. So Mondays from 11 to 12, but we can go a little bit longer if needed. So if we have a good discussion going, then obviously we'll keep going until 1 hour further. And tutorials will be live on Zoom, just like the Q&A sessions, every Wednesday from 11 to 1. And the tutorials are going to be very important for you because we will show you exactly how you can do each step in the assignments that I'll show you in a second. There's a quick note here on SPM that I introduced. So it's a MATLAB toolbox. MATLAB you know by now. I guess you've taken a course on it. And thanks to Jan for teaching MATLAB to you guys. I'd like to ask you to please install SPM12 before Monday or Wednesday, because on Wednesday we actually start playing with it already. So you can download it from the webpage that I'm showing here. I believe there's no link here, but this is basically how it looks like. Just type in SPM12 into Google and it'll get you here. And then Wednesday, we will give you the data to work with and you can start playing with the data already. Just as a brief overview, this is how it looks like. So you have your command window typically in the background. This is a Mac version of this. On a PC, I think it looks just a little bit different. So you have a number of buttons that I'm showing here. This is the pre-processing button. Let me see if I can change this to a laser pointer. There we go. So these are pre-processing steps. You have realignment, slice timing correction, normalization, segmentation, and smoothing. And again, we go through each of these steps in the tutorials. These are all required to prepare the data for a statistical analysis later on. Then you have your statistical buttons, if you will. Specify first level, specify second level. As I said, a first level is the basically brain-based or statistical parametric map for each individual subject. You have to compute this for every single participant, and then you move it to the next level, to the second level, to get a group level understanding or statistic that allows you to make inferences then, basically. Then you have your results button here, and you have some more advanced tabs like dynamic causal modeling, which we won't discuss in this course, and some display options that are shown down here. So display and additional functions, because you will eventually want to see and identify the statistics that you computed on top of the brain to illustrate where and how these activations occur in your sample. Okay, but this is stuff that you'll see over and over again on your own computer as well as in the tutorials, and this will become second nature to you by the end of the course. Here's the outline of the course. So the first lecture, which basically continues after this one immediately, is the Intro to Neuroscience methods. So we'll talk about different methods that are used in neuroscience and neuroeconomics, and also focusing in on fMRI and the fMRI data structure, which you'll already need to know to be able to handle the data. Then we move on to pre-processing in the second week. These are the first steps you do to prepare the data, to clean the noise that the scanner introduces, the noise that the person introduces, and see whether we can actually use this person in the data set afterwards. Then we have signal and noise, which is an aspect that you need to understand because it underlies a fundamental problem in fMRI research. We'll talk about experimental designs because that's really fundamental to get good data. There are timing issues here in the context of functional magnetic resonance imaging that you do not have when you run behavioral experiments like you just did in experimental economics or like you read about in behavioral economics. So we have some limitations based on the signal. So this ties in directly with signal and noise that we can counteract by better experimental design. Then we'll move on to statistical analysis, first and second level models. And finally, we have a lecture that sort of wraps everything up, talks a little bit more about second level models. And we'll also have a little bit of extra time for things that might be important to finalize your projects. But we might talk about brain stimulation a little bit. Okay. So my goal is that by the end of this course, after what you've just seen here on this slide, these are all the things we're going through. That's basically what the authors of this paper, or actually that's one of my papers, that's what we are talking about here. We're just describing what we've done. You will be able to understand this and you will be able to directly implement these kinds of things by the end of this course. So it's fundamentally something that makes you a better neuroeconomist, if you will, to be able to understand how these methods are used, what the strengths and weaknesses of these methods are, and you get first-hand experience with actual data. So I think this is a really exciting course, and I hope that you like it. I think you will. But obviously we'll work together on this throughout the next 8 weeks. Here is how we'll evaluate you. It's quite a straightforward and easygoing course, I think, in terms of evaluation. You'll have weekly assignments. So every week we'll give you an assignment. I'll talk about these in a second. They count for 45%. And they're basically exactly the things that I want you to go in, that all go into your final project. And your final project is a data analysis project and write-up. And the goal is to work through these computational processing steps on a real fMRI data set to basically, to get a handle on how to analyze fMRI data and to showcase this in a final project. Here are all the things that I've done in my weekly assignments, and then you write this up as a nice paper in your final project. You don't have to have programming knowledge, but it can help significantly. You can actually use what's called batch scripting. You can look it up in SPM, and this will allow you to basically work faster. If you're good at programming in MATLAB, you'll be faster than others. The data will be provided by us, and the data analysis will be done basically throughout the tutorials. Will help you with this. But you'll have to do some of your own work to prepare for it and to continue the work that you started in the tutorial. I see that there's something I didn't catch here. The lab is on Wednesdays, and it's taught by Jan Hausfeld, not Chi-Chung. This is from last year, so I apologize for this. Finally, here are your weekly assignments. Assignment 1 in week 1 is to simply read the paper that we provide you with and to describe the experiment that forms the data of that created the data that we're giving you. So the basis of the fMRI data you're going to be working with. What does this mean? So this basically means to read the paper carefully that we're giving you. It deviates just a little bit. It's a simple behavioral paper we ran to identify the best possible experimental parameters for the fMRI study that we ran subsequent to this paper. And I'll post a link in the lecture notes so you can find it, and I'll put it in the materials as well. In week 2, we're going to start going straight into pre-processing steps. So we'll do motion correction and slice timing correction, which are the first 2 steps in in most pre-processing pipelines. Then in week 3, we're going to look at, so this is the functional data that we're correcting there. We also have to look at the anatomical data that's collected. So we transfer the T1 to the EPI. The EPI is the functional data. So we're actually looking at 2 different data sets. One brain that's high resolution and one brain that has a high temporal resolution and continues on a timeline. So the EPI is the functional data set that's continuously recorded. We'll talk about this in a second. We'll go into co-registration here, then segmentation of the T1, which breaks down the T1, the anatomical data set, into specific subparts, including white matter, gray matter, cerebrospinal fluid, skull, and all the rest that's outside of the brain. And that allows SPM, the program, to align it. So the brain is basically, every single brain is recorded in a slightly different space, and subjects move throughout the scan. So that allows us to align it to a template that's called normalization. And this in turn allows us to then bring all the brains into the same space so that we can do statistics on all of the brains at the same time. In week 4, we'll go into design matrices, which means that is building your model, your statistical model, and potentially discussing additional regressors. And then finally, week 5, we're looking at contrasts, which are your first statistical analyses for first level models. And in week 6, we're going to do second level models and finalizing your final projects, but there's no assignment that comes with this. Okay, so this is basically to give you an expectation about the course. Just briefly, if you have any questions about this, don't hesitate to contact me or discuss it with me on Monday during the Q&A session. Good. I'll start right away on the next one, which I'll upload probably tomorrow.